Hello everyone, welcome back to my channel, and today I am going to talk about a very interesting topic that is on mutations. So in my previous videos I had dealt many of the genetic disorders. So let us see how these disorders are happening. That is all these genetic disorders and other sickle cell anemia, Tay-Sachs disease, Down syndrome. So we studied many of the genetic diseases. So all these genetic diseases are happening due to mutations. So mutation is one of the yet another important topic that we have to discuss. Yes, right now. Okay, come. Let us today learn about mutations. So what are mutations? Mutation means in biology, a mutation is an alteration in the nucleotide sequence of the genome of an organism. If there is any change in the nucleotide sequence of the gene, then mutation occurs. So, a mutation occurs when a DNA, that is when a gene, is damaged or changed in such a way as to alter the genetic message carried by that gene. If suppose if of a particular gene, if that has to code for a particular protein, due to the mutation, it will code for a different protein. So this is the result of mutation. This is the meaning of mutation. So what mutagen means, it is an agent of the substance which cause a permanent alteration to the physical composition of the DNA, that is gene. So mutagen is an agent that induces mutations in the DNA, altering the physical composition of the DNA so that the genetic message is altered or genetic message is changed. So let us see what are the mutations. Like base substitutions, deletions, insertions, like that. There are many types of mutations. So today let us discuss all these types of mutations in detail. Okay, first let us see what are these base substitution mutations. Base substitution mutations means a single base substitutions means that occurs in a point, at a particular point. Is known as a base substitution mutation, a point mutation in which a single base is substituted. Like example for that you can give is sickle cell anemia. That is a base substitution. Instead of glutamic acid, valine is formed. So that is due to base substitution mutation. So coming to the next is that, okay, so gene mutation means there are two types. One is point mutation, other one is frame shift mutation. So gene mutations, there are two types. One is point mutation. Point mutation means it is happening in a single nucleotide, change at a particular point. That is called as point mutation. And frame shift mutation means when a nucleotide or nucleotides, when a single or double. That is when a nucleotide or nucleotides, there is an insertion or deletion of a single nucleotide or two nucleotides, like that. So that is called as frame shift mutation. So gene mutations are of two types. Point mutations, which happens at a particular point with change in a single nucleotide, and frame shift mutations. Frame shift mutations when there is a deletion or insertion of a nucleotide or two nucleotides. That is the new frame shift mutation. Coming to the chromosomal mutations. So I had dealt chromosomal mutations, that is Down syndrome, Klinefelter syndrome, Turner syndrome. All these are the result of chromosomal mutations. How they are happening? Chromosomal mutations means which involves the changes in the structure or the number of entire chromosomes. So 21st trisomy, Down syndrome. There is an extra chromosome added to the 21st autosomal pair. That is called as Down syndrome. In Klinefelter syndrome, there is an extra chromosome added in the allosome pair. That is 23rd pair. The extra X chromosome added to the 23rd pair in Klinefelter. So that is an allosomal genetic disorder. So all those are the examples for chromosomal mutations. So mutations can also be classified as germline mutations and somatic mutations. Germline mutations means occurs in gametes. These are especially significant, germline mutations. These are especially significant because they are transmitted to the offsprings, and every cell of the offspring will have the mutation. So germline mutations are very significant because they are transmitted to the offsprings. Somatic mutations, they occur in somatic cells. So let us learn about silent mutations. What is this silent mutation? Silent mutation means they are the mutations in DNA that do not have any observable effect on the organism's phenotype. Phenotype means external appearance. So there will be no change. Though the mutation occurs, there is no significant remarkable observable effect on the organism's phenotype, that is external appearance. So they are also called as neutral mutations. Means even the mutation occurs, there is no change in the phenotype of an organism. So that is called as silent mutation. It occurs silently. So now let us see the types of another mutations can also be categorized into two types, like missense mutation and nonsense mutations. What are these missense and nonsense? Let us see. Okay, missense means, as the name itself signifies, missense means there is something different happening. So missense, there is a change in the nucleotide sequence resulting in a different amino acid sequence from the normal genome. There is change in the amino acid sequence. So that is due to the missense mutation. Change in the DNA base pair, that is, it is a substitution of one amino acid for another in a protein. So resulting in missense. So substitution mutation comes under this. Missense mutation, sickle cell anemia comes under, it is an example of missense mutation which is under substitution mutation. So in sickle cell anemia what happens? Glutamic acid is replaced by valine, that is adenine is replaced by thymine, resulting in a alteration in the amino acid sequence, thus resulting in the change in the amino acid, resulting in a defective gene. That is what happens in the 17 nucleotide of the gene for the beta chain synthesis of hemoglobin. There has to be glutamic acid, that is GAG, that there has to be GAG. Instead of this missense mutation, what happens? The glutamic acid is replaced by valine, that is GTC, that is the sequence. So thus the sixth amino acid in the chain becomes valine instead of glutamic acid. So this is about missense mutation. So let us learn about nonsense mutation. What is this nonsense mutation? Nonsense mutation means the protein chain synthesis stops abruptly because of the presence of stop codons, also known as nonsense codons. In my previous videos, I have explained about the stop codons and nonsense codons. Wherever this stop codons, that is UAA, UGA and UAG are the stop codons. Because of the occurrence of this stop codons or the nonsense codons, the protein synthesis will be abruptly ended in the defective protein, that is short. The protein chain will be shortened. So let us see, due to the change in the DNA base pair, there will be a shorter unfinished protein product will be formed due to the stop codons, that is nonsense codons. The protein synthesis stops abruptly due to the occurrence of stop codons, also known as nonsense codons. Okay, now let us move on to next thing, that is how do you identify DNA mutations? How to identify these DNA mutations? How they are occurring? These mutations, they exploit one or more of the basic properties of the DNA or the enzymes that act upon it. A single base mutation can be identified by any of the following methods, like DNA sequencing, which involves the identifying each nucleotide base pair in the sequence and comparing that to the normal gene. That is the direct sequencing method. We can see how the mutations occur. And now let us see on the transition mutations and transversion mutations. So transition mutation means when a pyrimidine is substituted by pyrimidine, that or when a purine is substituted for another purine, it is called as transition mutation. Transversion mutation means a purine being replaced by a pyrimidine or vice versa. So that is an example for transversion. So mutations can also be the result of the addition of a nucleotide known as insertion mutations or the removal of base pair, the nucleotide also known as deletion of mutations. So insertion mutations and deletion mutations. These are the kinds. Now let us see what is this back mutation. There is also known as back mutation. What is this back mutation? Let's see. What is this? A change in the nucleotide pair in a mutant gene that restores the original sequence and hence the original phenotype is restored back. Back mutation means due to the change in the nucleotide pair in a mutant gene, due to some change, the original phenotypic character will be restored. This is known as back mutation. So, diseases caused due to mutations. What are these inherited genetic disorders like cystic fibrosis, sickle cell anemia, Tay-Sachs disease, phenylketonuria, color blindness? All these are the examples of genetic mutations. Sickle cell anemia example is an example of transversion mutation which comes under substitution mutation. So, thymine is replaced to adenine for hemoglobin subunit. So, glutamic acid is replaced to valine, hence changing the beta subunit of hemoglobin. How do you test these mutations? Genetic testing looks at your gene level to check for any mutations. Test is done with a sample of blood, saliva, or with tissues. So, this is how disease can be diagnosed. And let us see about Down syndrome, how it happens. It is also known as 21st trisomy and it is known as non-disjunction during oogenesis. Down's syndrome occurs due to the non-disjunction during oogenesis. How is this happening? It is caused by an error in the cell division called as non-disjunction. Due to this non-disjunction, it results in an embryo with 3 copies present at the chromosome 21. There has to be 2 copies. This occurs in somatic cells. That is 21st pair. There will be, instead of 2 chromosomes, there is an extra chromosome added to the 21st pair instead of usual 2. So, hence resulting in Down syndrome. Prior to the conception, a pair of 21st chromosome is in either sperm or ovum fails to separate. So, this is the defect in Down syndrome what it happens. So, it is an autosomal disorder resulting in Down syndrome. So, all these things are due to the mutations. So, we have to know about mutations. So, I hope my video is helpful for most of you. Okay, then take care. Bye-bye.